Embark on a journey of inspiration and discovery with the Purdue Lecture Hall series, proudly presented by the Purdue Institute of Inflammation, Immunology, and Infectious Disease. Join us as we delve into the remarkable odysseys of these aspiring scientists, each crafting their own narrative in the world of science and groundbreaking research. Take a glimpse into their diverse cultural backgrounds and the journeys that brought them to Purdue University. Thank you for watching the Purdue Lecture Hall series. Remember to hit the like button and subscribe to our channel. Welcome, everybody. My name is Tommy Soares, Director of Scientific Strategy and Relations at the Purdue Institute of Inflammation, Immunology, and Infectious Disease. Welcome to another Purdue Lecture Hall series where we have the honor of welcoming today Sneha Subramanian from Claudio Aguilar's lab in biological sciences here at Purdue University. Welcome, Sneha. Thank you so much for taking time to tell us a little bit about your story, a little bit about your wonderful research. I know Dr. Aguilar's work happening now in his lab, and it's extremely exciting. Some really interesting biology and also some very interesting diseases that you guys are looking into. So welcome. Thank you for joining us.
Thank you so much, Dr. Soares, for having me today.
Absolutely. It's great. It's a big honor and our pleasure to welcome you. I'm going to, as typical, let you take the spotlight here. If you want, you can share your screen. I'm going to minimize myself so that you can be the center stage of the show, and you can tell us a little bit what's up. And we'll join you. I'll join you back again in a little bit. All right.
Thank you so much. So is the screen visible, like the title screen?
Yes, perfect.
Sounds good. Thank you. All right. So hi, everyone. I'm Sneha Subramanian, as Dr. Soares already mentioned. I'm a PhD candidate in the Aguilar lab at the Biological Sciences Department. And today I will be talking about my journey into pursuing sciences and also a little about the research that I do at the Aguilar lab. So a brief introduction about myself. I was born in Dayton, Ohio, in US, which is just 4 hours away by road from Purdue. And my parents are IT professionals. So they were working in the US, and then they returned back to India when I was only about 3 years of age. And we went to Delhi, which is the capital of India. So I was brought up in the Delhi NCR region, which is the National Capital Region. And I pretty much did all my schooling, my high school, my undergrad in Delhi, and I was there for 18 years. So now I just want to kind of give a brief glimpse of India. So these are some beautiful landscapes of India, and we have all the major geographical terrains, the desert, the coastal plains, mountains, the plateaus. So all the major geographical terrains, India is a house to all of them. And then I also wanted to highlight something about India, which is its extreme richness in heritage and culture. And it's actually said that India is a melting pot of cultures where a lot of diverse cultures coexist. And it's also said that every 200 kilometers that you travel in India, you can see the language and the culture changing. And some of these are just a few of the architectural marvels in India. On the left is the Meenakshi temple. One of the most notable features of the temple is that it has a hall of 1,000 pillars. And each of these pillars have intricate carvings on them with various mythological figures and scenes from the Hindu epic like Ramayana and the Mahabharata. And here on top left is the iconic Taj Mahal, It's one of the 7 wonders of the world. And it took 22 years to build this. And one of the most interesting facts that I like is that it has these 4 minars on 2 minars on either sides. And they're built such that they're slightly tilted outwards, so that in an event of an earthquake, they would fall away from the central tomb, thereby minimizing potential damage to the main structure. And here on the top right is the gateway of India. It's interestingly a ceremonial entrance it has been a ceremonial entrance for important guests and dignitaries visiting the city of Mumbai, especially during the British colonial period. And one of the most important or interesting facts about this is that it's a place of significant historical events, such as the departure of the last British troops from India in 1948, which marked the end of the British colonial rule in India. And in the bottom left is the Hawa Mahal, also called as the Palace of Winds. The most distinctive feature about it is its facade, which is adorned with intricate latticework windows or small balconies. And it stands as a testament to the architectural ingenuity of time. And finally, on the right bottom is called the Lotus Temple. It's renowned for its innovative and unique architectural design, but also for its role as a place for reflection, contemplation, and spiritual solace. So these are just some of the few architectural marvels in India, but I'm sure there's a lot, many more, and I'm probably just covering not even close to 1% of what India is and what it has to offer. On top of that, what also unites us is the variety of Indian dishes. And my friends from the US and other countries around the world are a testament to how much they love Indian food and its spices and flavors. So with this, I hope at least I inspired you to visit India and explore the diversity and the country, the diversity that the country is proud of. Okay, so moving on, this is where I talk about my journey to where it all began and where it started, which is my school, where I was there. I was there for 14 years, from kindergarten till my high school 12th grade. So I spent a good chunk of my life in this school, within these buildings. And the name of my school is Somerville School in Vasundhara Enclave, again in Delhi. And this is where my high school biology teacher, Dr. Pratima Lal, makes her entry in my life, who has been extremely instrumental in me ultimately pursuing biology and studying biology and doing research. I was actually amazed at the passion with which she used to teach, and something about her teaching just made everything seem very simple to understand. And in my 12th grade, we had a topic on reproductive biology. So she made it is of course a marvel, and the way she explained it was a cherry on the top. So she explained how 2 cells that are absolutely invisible to the naked eye can fuse together to form a baby. And so that was something which made me think a lot about how beautiful of a subject biology is. Simply topics like how processes in cells and human body are so tightly regulated, and how such intricate cycles and reactions in our body occur with so much ease and just by themselves. Like, it's not that we give instructions voluntarily from outside, it's all tightly regulated and it happens on its own. And then also other things like how the body or the organs, they work day in and day out only to keep you alive, how the heart beats, how the brain processes information, and I can go on and on. So this was like my first exposure to biology in so much detail, and she just made it sound like it was so easy and so easy to understand all these complex processes. And Another thing was that I'm also a very visual learner. So I turn to YouTube or different videos to understand these different concepts in any subject for that matter. And I came across this video by Nucleus Medical Media. And this is on YouTube. And it goes in depth, and the videos of in this YouTube video just explains all these processes with so much detail and so much depth that it's very easy to understand and grasp what's exactly happening. And in addition to that, I also came across this documentary on National Geographic called In the Womb, which again was a breakthrough in terms of how digital media can actually make learning so much easy. So this was kind of my turning point, where I was absolutely influenced and absolutely determined to ask the what and the why behind all the processes in biology, and especially the human physiology and human body. So next, at this point, I was sure that I wanted to pursue my higher studies in biology. So after graduating from high school in 2015, I got accepted into University of Delhi, where I pursued my bachelor's in zoology with an honors in Hindu College. So University of Delhi, I'm sure some of you would probably know that it's one of the world-renowned universities. And it's one of the institutes in the Institutes of Eminence. So the government of India provides a huge sum of amount to promote research and collaboration across different institutes in the world. And it's also part of the BRICS University League. And BRICS is short for Brazil, Russia, India, China, and South Africa. So all the top institutes from these countries have a university league, and that's called the BRICS University League. And inside, under University of Delhi, I did my undergrad from Hindu College, which is again one of the oldest institutes or oldest colleges in India, which was founded in 1899. And interestingly, Hindu College was a center for intellectual and political debate during India's freedom struggle, which served as a platform for famous personalities like Mahatma Gandhi. So my reason for why I pursued zoology honors was because I wanted to study all the topics related to biology, and I did not want to restrict myself to just a particular field, like, for example, biochemistry or only molecular biology or only cell biology at such an early stage of my education. And the curriculum for zoology honors had topics ranging from the animal kingdom, where we studied about each of the phylum. And then we had classes on biochemistry, molecular biology, cell biology, developmental biology, pretty much all the topics that you can cover that has got to do with the animal sciences field of study. So that's why I wanted to keep my area broad and basically learn everything that biology or animal sciences had to offer. So it was a pretty rigorous and comprehensive major to pursue, especially for someone like me who wanted to ultimately pursue research in the future. And this is where I met my mentors, who were absolutely responsible for my undergrad success. Dr. Late Prabir Sinha, Dr. Soma Gorai, Dr. Neetu, and Dr. Varunendra Singh. They actually really taught us the importance of scientific research. And they had the ability to ignite the spark of curiosity within us and also keep it alive. And they highlighted the wonders of research and science. And their passion and dedication to teach with the excitement of discovery was absolutely amazing to me. And that really inspired me further to and be absolutely sure that I wanted to do research after this. And all my 4 professors actually guided and encouraged me throughout my grad school application process. And because of their unwavering support, this is where I am. And so I'm absolutely grateful for their support throughout the 3 years from 2015 to 2018. And they still support me and they still are in touch with me. And they are always in touch with the research that I'm doing right now. So I keep updating them about my research and my successes as well. So within these 3 years, from 2015 to 2018, I actually also did internships in each of the summers. So my first internship was a volunteership or a teaching internship in summer of 2016. So I went for a volunteer program through the organization called ISEC, which is an international youth-led organization focused on leadership development and cross-cultural exchanges. And my project was in Miami. And so ISEC in Miami at Florida International University was hosting us. And this is a picture of the students who had come for the volunteership, and students from different countries like Tunisia, Turkey, France, Colombia, Mexico, Kazakhstan, India had come to work on the projects. And I specifically worked with the organization called Touching Miami with Love in the Overtown area. And this particular organization aims to address issues that's related to socioeconomic challenges, education, and community development by fostering a positive impact on the lives of those that they serve in the community. So my role was to teach a class of children around the age of 9. And I designed coursework and taught the children science and visual arts over the course of 8 weeks. And this picture right here is my very enthusiastic, energetic class of 9-year-olds and also my colleagues. This experience was actually a game changer for me personally as well as professionally. Personally, because this was my first time I stayed away from my parents, since even for my undergrad studies, I was during the courses and my undergrad, I was a day scholar and I would travel from home every day. So leave alone going to a different place in India, I traveled about 8,000 miles alone and lived by myself and immersed myself in a different new culture altogether. So that was a very life-changing moment for me. Professionally speaking, this experience allowed me to communicate complex concepts in an engaging and simplified manner, which I think is a very critical skill required in science education. Where clear communication of complex ideas is crucial. So this was my first-hand experience where seemingly unrelated experiences contributed to my overall growth and skill development. So moving on, I did 2 more internships. The first was in summer 2017, which was a research internship. And for this, I got selected actually by the prestigious Indian Academy of Sciences, IAS INSA NASI Summer Research Fellowship. And this is a fantastic program where the primary objective is to provide young undergrads and grad students with exposure to research in various scientific disciplines and to encourage them to pursue careers in science and research. So I was selected to do my research in Mysore in Odisha in the molecular genetics laboratory under the guidance of Dr. Devasmita Alone. And here I was studying the molecular genetics of Fuchs endothelial corneal dystrophy. And here I learned a lot of basic skills. So I learned skills like extracting the DNA, genomic DNA from patient blood samples, polymerase chain reaction, the PCRs, and I learned how to do Sanger sequencing and also restriction fragment-led polymorphism. So this experience was my first ever hands-on experience in a research setting or in a research lab. Then the next I did was another research internship at Regional Center for Biotechnology in the RNA Biology Laboratory of Dr. Gitanjali Chawla, who actually was at that point shifting her lab from IU Bloomington. And I studied the role of microRNAs in aging and maintenance of neuronal integrity. This was a different experience since this was the first time I worked with a model organism and to see which is the Drosophila melanogaster or the fruit fly. And to be able to see the common fruit fly up so close under a microscope itself was fascinating for me. I would dissect the Drosophila brain, fat body, and we were looking at microRNA MIR125 and LET7, and also looking at the target of these microRNAs called the KINMO protein. So we wanted to see if the elevated levels of KINMO was responsible for age-related neurodegeneration. And the next thing we also wanted to look was to see the effect on the lifespan of the flies when they're fed with food that's ad libitum, or when they're fed a dietary restricted food, which is either protein or calorie deficient food. So I learned a lot of different skills, which of course was to breed, handle the fruit fly. And then I also used the GAL4 UAS system, which is a popular molecular biology tool, which is used to control gene expression, where the GAL4 protein activates target genes that are positioned downstream of the UAS, or the upstream activating sequence. Then I did dissections under the microscope, and then immunostaining, and then confocal microscopy. So the next, once I was done with my undergrad, I was looking for options to do next, which was grad school. And I applied to very few schools actually, because I had done a 3-year undergrad degree instead of the conventional 4-year undergrad. So for that, my criteria was looking into schools and programs that actually took students with a 3-year undergrad. So many schools do consider a 3-year undergrad with an honors degree with equivalent to a 4-year undergrad. So I researched into that, and Purdue was one of my top schools of preference, since some of my family members already were studying at Purdue, and at that time, and we had heard only good things from them about Purdue. Whether it was cost of living, whether it was the diversity on campus, or the research opportunities and the cutting edge technology that Purdue had to offer. So, and then fun fact, I actually applied to Purdue Biological Sciences only for a master's degree, because in India, and I'm pretty sure even in Europe, the logical step to do, like after an undergrad, is to do a master's instead of doing a PhD. So I was absolutely unaware of the fact that you could do a PhD after just your undergrad. So I just applied for a master's, and when I was being interviewed by Dr. Nancy Palace here in the department, I told her that I want my master's to be very research extensive. So that's the point when she told me that you could actually do a PhD without a master's directly from an undergrad. So that seemed to be a very good option for me. And I just changed my degree goal from master's to PhD. And finally, I got accepted into the Department of Biological Sciences. And I started in August 2018. And this was the time I made my journey back from Delhi to the US, or as they call, time to go back home, as the immigration officers say it. And finally, I had a stop over at London when I was traveling from Delhi to Chicago. And the immigration officer was like, "Oh, going back home." And I was like, "Oh, yeah, after 18 years, I guess." So yeah, I made it to Purdue. These are some of my pictures actually from my first year here at Purdue. This is the cohort, the 2018 fall 2018 cohort. And here in the bottom is my first class that I TA'd for, which is, I think, 204 biolab. So the next question was, we all had to make an important decision, which is which lab to join. And actually, while I was writing my statement of purpose, I had done some research on the faculty and their work in the biological sciences department. And I came across this video of Claudio, Dr. Aguilar, where he spoke about the first targeted delivery agent against bladder cancer that was developed in the lab. And this video actually captured my attention, and that made me really interested in his lab itself. And when I met him during the orientation week, so biological sciences has this one full week of orientation where faculty from different areas or different majors in the department come and talk about the research that they are doing in the lab. So Claudia had also delivered a lecture on that, and that again further strengthened my decision to join the lab and rotate in the lab and then potentially join the lab. So at that point, when I met him during the orientation week, I could see everything that I had expected in a mentor in him. And so throughout the year, since we're supposed to do 4 rotations in different labs, his lab was obviously one of the labs I rotated in, and I actually ended up doing 2 of my rotations of the 4 in his lab, and then ultimately joined his lab in summer 2019. And at this point, so one of the biggest factors for me to choose a lab was, of course, if the research aligned with my interest or not, but also the lab environment. And one of the biggest factors was the advisor's mentorship and approachability. So Claudio always had an open door policy and is always approachable and reachable whenever you have any question, either in person or email. He's always reachable. So that is something I wanted because I did not have like too much research experience prior to this. So I wanted someone who's pretty hands-on and is always there to answer my questions. And Claudio had that in him. So that was an easy decision for me to make and join his lab. So this study that he was talking about, and which inspired me to join his lab, got published in the International Journal of Cancer and also made it to the cover of the journal. And this is the QR code if you want to scan and look into this paper further. And in summer 2019, when I joined the lab, I actually got the opportunity to work on a follow-up project, which was the 2nd generation of targeted therapy against bladder cancer that was again developed in the lab, which used a ligand-fused anthrax toxin chimera. And this study also was published in International Journal of Cancer. And it was absolutely a great honor for me to be a part of the study because of the high impact that the study ultimately had and the high media news coverage that this study had. And we again made it to the cover of the journal. And this is the QR code to the paper in case you're interested. Okay, so now I'm going to share my work on one of my projects, which is more in the basic research kind of area, as opposed to my other projects, which are very translational in nature. So before I delve into my work, I want to give a brief background pertaining to it. So the protein that I'm looking at is the epidermal growth factor receptor. So it's a transmembrane protein. It has an extracellular domain, a transmembrane domain, and a tyrosine kinase domain, which is the intracellular part of the protein. It can exist in 2 conformations. One is the tethered conformation, where the subdomains 2 and 4 are kind of folded onto each other, whereas the second conformation is the extended conformation, and this conformation comes into play when a ligand has to go and bind to the receptor. So the receptor basically binds between domain 1 and domain 3. And then ultimately, these receptor molecules, they dimerize. So another copy of the receptor comes and they dimerize. And they do something that's known as the cross-phosphorylation. So they phosphorylate each other. And there's a downstream cascade, or like there's multiple phosphorylation events that occur in the intracellular domain or the cytosolic tail of the protein. And this is kind of the beginning of what we call as the signal transduction or the cascade pathway. So this phosphorylated cytosolic tail is responsible for recruitment of multiple proteins, such as the SOS, which is son of 7-less, or the GRAP2 protein, which then and this GRAP2 SOS complex then activates the RAS protein by allowing it to attach to the GTP molecule. And then as a result of that, it leads to a cascade reaction. And this pathway is known as the MAP kinase pathway. And the other pathway that's also activated by this is the PI3 kinase pathway. And broadly speaking, these 2 pathways are primarily responsible for proliferation and survival in a cell. And this whole process in a nutshell is called signal propagation. However, as any other process in a cell, there needs to be a balance to how much signal propagation can happen and how much signaling can happen in a cell. So to balance that effect, there's something known as signal termination, which is to stop the signaling event. And this happens by the recruitment of a protein called CBIL, which binds or recognizes the phosphorylated cytosolic tail of the protein. And this CBIL protein is a ubiquitin ligase, or basically it tags the protein with a molecule called the ubiquitin, which is then read as a signal by the cell for endocytosis or internalization of these molecules, these receptors into the cell, and then ultimately leading to lysosomal degradation. Now, these receptor tyrosine kinases can have different mutations in them, such as amplification or different stop mutations or different frame shift mutations, which ultimately are responsible for the formation of tumor due to really high levels of proliferation and survival of the cells. So now I'll briefly go over what are the different abnormalities of the EGF receptor. So the first is amplification. So amplification, as the name kind of suggests, it's just more number of copies of the receptor. So in a normal cell, if you see, let's say, one receptor, hypothetically speaking, in a cancer cell, you'll see multiple of these receptors, like probably 10, 5, 10, many copies. So as a result of this, the downstream signaling that happens due to these receptors is exacerbated. So there's a lot of downstream signaling happening and a lot of proliferation, a lot of survival of the cells. Another abnormality is the mutation. So mutations can be of different types, and the effect of the mutation can also be of different types. So the first is endocytosis deficient. So what happens is, in one of the kinds of endocytosis deficient mutation, there is the phosphorylation of the cytosolic tail is insufficient, and as a result of which the CBIL protein does not get recruited. So if you remember a few slides ago, I mentioned the CBIL protein is responsible for signal termination, and that recognizes phosphorylated cytoplasmic tail of the protein. But if that's insufficient, then CBIL does not get recruited. And as a result of this, there's no endocytosis, no degradation of the receptor. And the receptor is pretty much stuck on the plasma membrane. So it's constantly signaling because it's stuck on the plasma membrane. Other type of mutations could be one which is constitutively activated. So for signaling events to occur due to these receptors, traditionally you need the ligand to go and bind to the receptor, and then that results in cross-phosphorylation and then downstream signaling. But in mutations, some of these mutations, what they do to these receptors is that they don't really need the ligand to go and bind to the receptor to be able to start the cascade. They are just active by themselves. And so essentially there's nothing that's regulating their activity. So it's always in its own position. It's always signaling. So one of the mutations is the L858R, very commonly seen in lung cancer. And the other mutation, which is what I'm going to be talking about today, is the EGFR-V3, or the epidermal growth factor receptor variant 3. And as you might have expected, because they're constitutively activated, there is exacerbated downstream signaling. So EGFR abnormalities are very common in cancers. And these are, in a nutshell, these are like all the different cancers where you can see abnormalities with the receptor. And lung cancer and glioblastoma multiforme, or the brain cancer, are the two major cancers where you can see these abnormalities. So next, now I'm going to start sharing some data regarding my research for this particular project. So we were very interested in exploring these EGFR mutations, specifically patient mutations, or mutations that are seen in patients, and to see how these mutations traffic in a cell, or what their localization is in the cells. So for that, I made these constructs, a lot of biotechnological molecular biology tools, and they have a reporter molecule called the green fluorescence protein. So I have the EGFR protein, the mutated or the wild type, and then that has a tag on it, which is the green fluorescence protein. So when I introduce these constructs, or the DNA, it's called transfection. So I transfect them in cells, And then ultimately, I can see my protein of interest under a fluorescence microscope because it has a green fluorescence protein attached to it. So I introduce them and then I remove the serum. So when I say serum starvation, it means removal of any growth factors. So I remove the growth factors and I see that after I transfect the cells and let them transfect for about 12 hours, remove the serum or remove the growth factor, I see that my protein gets concentrated on the plasma membrane. So it forms like a boundary or like an outline of the cell. And this is because now my receptor is all concentrated on the plasma membrane and it's waiting for the ligand, or the EGF or the epidermal growth factor ligand to come and bind to it so that it can then start the signal transduction and then get internalized and endocytosed. So it's basically hungry for food in a way, if you put it in a layman term. And then once I serum starved, I see this, what I did was, so I had multiple mutations. So one of them was EGFR wild type, which is just the normal EGF receptor found in normal cells. The other was the K721M. So this is a mutation which is deficient for endocytosis. And it's a mutation in the cytosolic tail. And the second is the L858R, which you might remember is the constitutively active mutant, and also EGFRV3, which is again the constitutively active mutant. So then after that, after I serum starve them, I add the serum to this, and I see that these receptors get internalized and form these punctate structures or these kind of tiny dots, which are basically, telling that these receptors are now inside compartments called endosomes. And I saw a similar effect with L858R, but for K721M, I see they're still kind of stuck on the plasma membrane. You can probably see some of the outline of the cell, which is very in line with the fact that they're endocytosis deficient. But the turning point of this project was where for EGFRV3, we saw these huge intracellular compartments, which were previously unknown or never noticed. And these are very bright structures, and we always see them every time they're under the influence of serum. So this drew our attention, and we wanted to pursue this furthermore and try to see what's forming these compartments, what is the nature of these compartments, so on and so forth. So before I show the data of the next few studies, I just want to kind of orient everyone to know what exactly EGFRV3 is and how that's different from EGFR wild type. So this is our wild type receptor, as I showed a few slides ago. The V3 mutant is the EGFRV3 is a much smaller protein. So it has a huge truncation in the extracellular domain. So it's lacking a huge chunk there. And it's an in-frame deletion. And so it's a mutation, which is an in-frame deletion of exons 2 to 7. And with the deletion of these exons, there's also a substitution, which is the introduction of a novel glycine. So some characteristics about this receptor, it's found overexpressed in prostate cancer, glioblastoma, head and neck squamous cell carcinoma, and breast cancer. And it's constitutively active, and it's also amplified. So you have also multiple, not only is this receptor mutated, it's also amplified. So you have multiple copies of this receptor. And the best thing about this is that it's only found in tumors and it's not found in normal tissues. Therefore, ultimately, when we look at this project, we want to generate targeted therapies against this target. So it's going to be a little easier for us because it's only seen in tumors. So thereby we should not be able to, we don't have to worry about the non-specific targeting or the chances of it targeting normal tissues, because normal tissues don't have EGFRV3 in them. And then binding of the EGF ligand has been disputed. So because it's lacking the ligand binding domain. So some studies suggest it binds, some studies suggest it doesn't bind to it. And then some other studies, they have also shown that EGFRV3 can heterodimerize with the wild type receptor. So this is just to kind of recap. So we see these structures. And so next we wanted to look at the kinetics of these compartments. We wanted to see how early on do these compartments start forming. And so at 0.5 hours or at 30 minutes after serum addition or after addition of growth factors, we still see they're pretty much plasma membrane accumulated. So they form the outline of the cell. And about 4 hours later, we see that that we don't see the outline of the cell anymore. So it's definitely getting internalized now. And we see it's mostly around the nucleus. So DAPI, or the blue stain, stains the nucleus of the cell. So we see a lot of green stain around the nucleus of the cell. And then by 8 hours, we start seeing these bubbles, or like networks, as we like to call it, around the nucleus. So this is kind of like the time point where we start seeing their emergence. And by 30 hours, these are fully formed structures, very discrete in nature. So around 30 hours, we see them fully formed. So I quantified them. And so as the duration of stimulation with media increases, the number of these compartments per cell also increase. And these compartments also assemble mostly at lower total fluorescence intensities, which suggested that they're not an artifact of overexpression, but it is actually something that you see even at lower total fluorescence intensities. So next, we also wanted to kind of see what component of the serum, because serum is a concoction of multiple different growth factors. So we wanted to see if we cherry pick, we wanted to see which growth factor or if there's more than one growth factor, which of them those growth factors contribute in the formation of these structures. So we chose EGF. And we saw that there was a dose dependency. So with increase in the dosage of EGF ligand, we see the number of compartments increasing. We also chose the PDGF ligand, which is the platelet-derived growth factor. And we don't see any structures there, which suggested that not any growth factor can trigger compartment assembly. And this also suggests, this experiment also suggests that the EGFR-V3 traffics through the plasma membrane and then internalizes upon addition of EGF, and that EGF ligand is required for compartment assembly. Next, we wanted to further just confirm that these structures are indeed a product of EGFR-V3 getting internalized in the cell. So what we did was to make a mutant to create a mutation or a double mutation. So we had EGFR-V3, but we also mutated the lysine residue at 721. And so we made the K721M mutation. If you remember, the K721M is the endocytosis deficient mutation. So we introduced that in EGFR-V3 so that we can interfere with the internalization of EGFR-V3. And we saw that just EGFR-V3 shows its characteristic compartments. But when we have this mutation in EGFR-V3, we do not see the formation of these compartments. So this suggested that internalization and or signaling events that are mediated by the lysine 721 residue are important for compartment assembly. And one of the future directions here we would like to pursue is to see the effect of endocytosis inhibiting compounds such as pitstop2 or dinosaur, which also performs these similar functions of inhibiting endocytosis. Okay, so before I go into some more pieces of data, I just want to quickly go over what's a receptor-mediated endocytosis. So typically, the ligand will bind to a receptor which is present on the plasma membrane. Once it binds to the ligand, it triggers the internalization. And when I say internalization, it's also synonymous with endocytosis. So it gets incorporated into these vesicles. So the membrane kind of folds inwards, and then these vesicles are formed. And as a result of this, it invaginates, and then it gets inside the cell. So basically the ligand, the receptor complex is now inside a vesicle, an intracellular vesicle. And then these intracellular vesicles, they're also called endosomes, and they can be either early or late in nature based on the time points that they are in the trafficking or the endocytic pathway. So the first is early endosomes, and EA1 and RAB5 are some proteins that are characteristically found only in the early endosomes. So they are called protein markers. So a protein marker for early endosome is EA1, which is the early endosome antigen, and then RAB5. And then further, the vesicle then matures into what's called as the late endosome or the lysosomes. And then the receptor, the ligand goes for degradation, where LAMP1 is a marker for lysosome, and then the receptor is recycled back to the membrane again to repeat the cycle. So that's endocytosis. And there's also something known as the secretory pathway, where the protein is made and then it's sent, packaged, and then sent to where it's destined to be present in the cell. So the cell, so a DNA is translated into protein. Proteins are synthesized in the endoplasmic reticulum, where PDI-A3 or protein diisomerase is a protein marker for the ER. And then it's then packaged in the Golgi network. So TGN46 is protein marker for trans-Golgi network, and GM130 is a protein marker for the cis-Golgi network. And then proteins are then sent to where they're supposed to be based on particular sequences that these proteins have. So this is a very brief overview of what the endocytic and the secretory pathway looks like. Now, using this information, I'm going to show you the rest of my data. So we wanted to find out the nature of these compartments. We wanted to see what nature these compartments have when you look at the different compartments in the cell. So what we did was to do a co-localization experiment. So we did immunostaining. We stained the cells that had EGFRV3 in them, and we stained them with these different protein markers that I just mentioned. So the first one was EA1, which is the early endosome marker. And we see that there was an absolute correlation, like absolute 100% co-localization of the EA1 marker with EGFRV3, suggesting that it has early endosomes have a role to play in the formation of these compartments. Next, we also stained with LAMP1, and then we stained with GN130 and TGN46, and none of these markers showed any co-localization with EGFRV3. Then we also quantified this, so just not qualitative, but also quantitative analysis. And we saw that there was almost 96 to 98% co-localization percentage between EA1 and EGFRV3. So this definitely suggested that early endosomes had a role to play, but we wanted to go one step further and confirm the role of the early endosomes. So for that, I co-transfected EGFRV3 along with M-cherry, or a red fluorescent RAB5 wild type. So RAB5, if you remember, is another early endosome marker. And when I co-transfected them, I see again, when we merge them, I see that there is again a one-to-one correlation of both these proteins. So this definitely strengthened the fact that there is an early endosomal nature to these compartments. So next, what we also observed actually during kinetics experiment that I just showed a few slides ago was that on certain time points, we were seeing these compartments, or like these bubble-like structures, which are very continuous with the nuclear envelope. And this made us think if the endoplasmic reticulum has a role also to play in this, because the endoplasmic reticulum is very continuous with the nuclear envelope or the nucleus. So what we did was to stain with PDI-A3, the ER marker, and we see that when we co-stain with EA1 and PDI-A3, both these markers co-localize with EGFRV3. So this suggested that these compartments have a mixed ER and an endosomal nature to it. So this brings me to my working model, which we think, based on a lot of other studies from other groups and based on our data, this is the working model that we have right now. So we transfect EGFRV3 and that gets synthesized in the ER. So there's EGFR-V3 that we see that's accumulated on the ER. And these tiny molecules are my representation of EGFR-V3. And then that goes through the secretory pathway. It gets packaged in these buds off from the ER, gets packaged, gets sent to the Golgi in these vesicles. And then from the Golgi, it then reaches the plasma membrane. And then in the plasma, once it reaches the plasma membrane, upon addition of growth factors or due to other receptor tyrosine kinase, there could be multiple reasons. It gets internalized through the endocytic pathway. And then the first compartment it reaches is the early endosomes. And then early endosomes, as I mentioned, has RAB5 and also EA1 present in these compartments. And these studies have actually also shown that RAB5 is present in the endoplasmic reticulum as well. So to recap, we have RAB5 that's present in the early endosomes as well as in the ER. And this study, and it's a very extensive study, especially the paper by Friedman et al. They have done super resolution microscopy and cryotomography to actually visualize that the endosomes actually dock into the ER, and that basically that the ER and the endosomes form contacts, and there's like a web-like structure around of the ER around the endosomes. And this is the piece of data that also shows that, and this is our piece of data where in the inset here, you can see these tiny EA1 puncta or these tiny EA1 dots are actually surrounding or getting recruited during the compartment assembly. Okay, so, and another piece of information that we got from these studies was that EGFR is responsible for activation of RAB5. So now we have EGFR that's present in the early endosomes and also present in the ER. And this EGFR is responsible for activation of RAB5. And we ultimately see fusion. So the function of RAB5 is to fuse membranes. That's the function of RAB5. And what we speculate is that due to the activation of RAB5, because there's EGFR present in the early endosomes and ER, RAB5 is activated and then it does its function, which is to fuse. And because the study shows that endosomes and ER contact, we suggest that these endosomes are fusing onto the membrane of the endoplasmic reticulum. And these are the compartments that then ultimately bud off to form a single entity. So what I also wanted to do was to be absolutely sure that this is what in a way is happening. So I wanted to interfere with RAB5's function of fusion. So what I did was to use a construct called RAB5 S34N, which is a dominant negative mutant of RAB5. So it does not work the way RAB5 is supposed to do. And I wanted to see the effect of this RAB5 dominant negative on the formation of these compartments. And I saw then in the cells that had RAB5 S34N as well as EGFR-V3, I saw that the number of cells with both these constructs showed much less number of V3 structures than the cells that did not have RAB5 S34N in them. So this definitely also further confirmed or further strengthened the view that activated RAB5 is needed for EGFR-V3 compartment assembly. So one of the future directions would be to perform super resolution microscopy and cryo-electron tomography to be able to see these structures at really high magnification with high resolution. So this is all the ongoing stuff that I have. And I am just going to quickly go over the summary, just a few take-home messages from the work that is ongoing right now. One is that these EGFR-V3 compartments assemble into previously unnoticed large intracellular compartments. So EGFR-V3 basically forms these compartments, and the assembly of the compartments depends on different factors. One is the presence of EGF or growth factors. And it also depends on the internalization or endocytosis of EGFR-V3, as well as on RAP5 activation. And the nature of these compartments is that they have a mixed endosomal, early endosomal, and endoplasmic reticular nature to them, and that these compartments are unique to EGFR-V3. And then finally, EGFR-V3, what we speculate so far based on our data and also other studies is that EGFR-V3 might be enhancing the fusion of early endosomes to the surface of the ER. With this, I'd like to thank my lab and then my research committee, Dr. Claudio Aguilar, Dr. Tim Ratliff, Dr. Angeline Lyon, and Dr. Jason Hanna, who meet with me every year to go over the progress of my project and then give me really good inputs on what direction to follow, what experiments to do, what are the achievables. And then I also want to give a shout out to my undergrad, Anna, who helps me take a lot of these images and analyze them as well. And finally, I would also like to thank the funding agencies, the Purdue Institute for Cancer Research, Purdue Institute for Drug Discovery, as well as the NIH. With this, I'd like to thank you for listening to my talk. This is my contact, my email ID, and this is my LinkedIn in case you're interested in connecting. Thank you.
Awesome talk and such wonderful research. Thank you so much for taking time to tell us a little bit about it. Tell us a little bit about the adventure. I'm going to ask you maybe if you can stop sharing your screen, and like that we can get back onto side by side. Sneha, what wonderful work, and so exciting to see so much progress too. Wow, you've been working really hard, I can tell. And you've been doing a lot of experiments, I can tell. It's great though, really cool. What's been your favorite part so far
about research?
Yes,
I think it's when you do an experiment, especially with biological sciences, the experiment mostly spans across a few days. So whenever I start an experiment, I'm like always excited to know the end result, to see what I'm going to, if it's going to be, the results I'm going to see are going to be in line with what I'm expecting, or if there's something new I'm going to discover. So that's one of the things that keeps me motivated and keeps me going forward. So that's one of the most exciting things about research, I think, so far.
I agree. I think, there is so much to be seen on the other side of the experiment, in the analysis of the data, to see if you really got something, if it's really something new, something novel, something that you thought could be even possible. And it never is. Really, we're always surprised by the stuff that we get, especially, in such a forward field as this, endosome trafficking and what's happening inside the cell, and still being able to crack that inside, and having so many fun technologies at your hands here at Purdue University where you can start asking those questions. Wonderful. Tell us a little bit, what's next for you? What are you thinking? Where are you going to go after this, or where are you going to take this research?
I think I'm right now interested in working in the industry. So a lot of my projects, the ones that I didn't talk about today, are very translational in nature. So it's a lot of bench to bedside kind of research. So, and I really think that it's like cancer is a global health issue. So I really think there's a lot that has happened, but a lot of research is still left to be done. I think I can really contribute in developing some therapies, some novel therapies that can actually help lives of patients and improving quality of their lives. So right now, I think I see myself working in an industry like a pharmaceutical company or a biotech company.
I am certain that many who are listening to this program are going to be inspired by those true words and real words of, wanting to make a contribution, wanting to save people's lives and do something that is practical and applicable. Thank you so much, Sneha Subramanian, for telling us and showing us. And my gosh, what a journey back and forth. It was awesome just being part of it and listening to it. Thank you so much for letting us.
I wanted to say thank you so much for doing this, actually, because I was looking at all the pictures and it was a fantastic stroll down the memory lane. So I think it was a great opportunity for me to kind of recap all the years that have gone by.
Wonderful. And that is also part of this, right? I get that from a lot of the lecture hall lecturers. They say, wow, I realized how much I had done and how far I have gone just by doing this type of reflection. And so I thank you for that comment. And I hope, again, like people do realize that you're contributing a lot and your work counts. You are a pioneer in your area, and it's an honor to have you share some of that with us today. Thank you, Sneha. And have a great afternoon.
Thank you, you too.
Thanks. Bye-bye. Thank you for watching the Purdue Lecture Hall series. Remember to hit the like button and subscribe to our channel.